Hi you guys, I have another mini lecture for you on proteins, and you also have some videos to watch that are not mine also, but I wanted to guide you through this a little bit. So proteins have a structure that varies a lot. So I'm telling you here, proteins have a more variable structure, and they come in very different shapes. So in this slide, you can see that it can be a structural protein, which means it forms the structure of something like collagen. And you've probably heard of collagen, right? It's in your skin and it's giving it structure and shape. It could be something like a contractile protein. These are muscle proteins that are allowing the muscle to contract and to relax. It could be one that is in charge of transport through a cell or storage, or they could be enzymes. So the variability of these molecules is huge. So when thinking of what that molecule looks like, I have another video there where you're recognizing molecules. We're going to look a little bit at the structure here. So the monomers of proteins, so the word monomer means the smallest unit. So the smallest unit of a protein is an amino acid. And you know about amino acids, you have them in your diet. You need them in your diet if you're going to manufacture all the proteins that you do in your body. So what does an amino acid look like? This is a picture here of an amino acid. It will always have an amino group if it's an amino acid. What's an amino group? A nitrogen and two hydrogens, NH2. Okay? So always will have an amino group. What else will it always have? It'll have that central carbon, and attached onto that central carbon, it will have a carboxyl group. Carboxyl group, a COOH. So that's your recognition. You look at this, you see, oh look, I see an amino group. Oh look, I see a carboxyl group. I have no idea what amino acid it is, but if I have those two, I know that I'm talking about an amino acid. This R up here means that attached onto this may be a variety of different atoms, and that's going to make up the 20 different amino acids depending on what's attached on to this R here. So they'll all look a little different. Okay, but what you'll be recognizing is the amino and the carboxyl group. So that's the general amino acid structure. This is an example of some of these different ones. Not that you would memorize this, but this is glycine, and it would have NH2, COOH, and only an H in that location. This is cysteine, NH2, COOH, and then something different. And it's not something you're going to memorize, but it's something else. And you've already recognized it as an amino acid. At this level of a course, that's all you need to do. Next would be tryptophan. So in tryptophan, and this one you find in soy, in eggs, in things like spirulina, those would also have NH2, COOH, and a more complex ring here. And you don't have to worry about that at all. Okay, all you know is that you've recognized it as an amino acid. I have made a different video on dehydration synthesis when I used the models that we use in chemistry. Here it is written out for you, so I want you to see it. If you had now dehydration synthesis done on a protein, because I demonstrated it on a carbohydrate, you would have an amino acid with its OH, another amino acid with its H. If we're going to join these two, we're going to lose water. So it's going to be dehydration synthesis, because you're going to drop an OH from one, drop an H for the other, forming water, H2O, and now I have joined these two monomers, smallest unit, into a dipeptide. So now it's not a disaccharide, it's a dipeptide when we're talking about a protein. Okay, and the connection between those two is called a dipeptide bond, or a peptide bond, sorry. Okay, The next thing I want you to know is how the shapes of these proteins vary. So if you imagine them linking these amino acids in one long, simple chain of amino acids, you're going to have a very simple structure. And that's going to be called the primary structure of a protein. And it's indicated here. You have one big, long chain as you made a peptide bond, peptide bond, peptide bond, and you chained them all together. However, some proteins are not as simple as that, and they'll do things like they'll coil, or maybe they'll pleat in space. And then we've gone on to a second structure because it's sort of three-dimensional, a different shape, right? It's three-dimensional now, and that structure is called secondary structure. So in this diagram, you have one example of it coiling and one example of it pleating. Secondary structure of a protein. What would be tertiary then? The tertiary structure is, imagine you are coiling something and you coil it so strongly that it actually twists over itself. So you've probably done that with a string. If you twist, 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 it'll end up curling over itself. And that would be the tertiary structure of a protein. How do you get to the last one here on the list, which is quaternary structure? A quaternary structure then is getting one of those tertiary structures, they're already coiled up, and getting 4 of them together and bonding those. So when you bond 4 of those together, you have a quaternary structure. You had to have had the tertiary first. And those are the different shapes of proteins. I'm going to stop it there. Thanks.